Good morning, everyone. Can we take a seat? And we are still on time. I think we ended perfectly at 10 o'clock this morning. We already have a great session and with many valuable sharings. Then it's my pleasure to introduce the speakers for the second part of session one. As mentioned earlier, it's going to be a niche area for locally acting GI tract PBPK modeling. The first speaker will be Dr. Sherin Thomas. She's a pharmacologist in DQMM, Division Quantitative Method Modeling, ORS OGD. Her presentation will be combined with Dr. Nikoletta Fotaki. He's the professor of biopharmaceutics, CTI, University of Bath. With that, and then we'll have the third speaker, Harshil Shah. He's the Senior Director Bioequivalence, COSAT Pharmaceutical Inc. And with that, I want to welcome Dr. Sherin Thomas to open up with the first talk. Thank you.
So for my talk, as Liang mentioned, there'll be two parts. The first part I'll be covering, and the second part Dr. Nikoletta will be covering. So the topic of the presentation is integration of biopredictive dissolution and PBPK models for the evaluation of GI locally acting products. And I'm Sherin Thomas from DQMM. So the usual disclaimer, this presentation reflects the views of the presenter and should not be construed as to represent the FDA's views or policies. So for GI locally acting products, demonstrating bioequivalence is a bit challenging because these products tend to reach the site of action before they enter the systemic circulation. Hence, the systemic exposure may not usually reflect the drug concentration at the site of action. So in general, B recommendation for oral locally acting GI drug products are based on the drug product properties and the mechanism of action. In addition, PBPK models can also be used to support the B evaluation of such drug products. You can use the PBPK models to explore the correlation between the dissolution in the GI tract and the systemic concentration of these products. So for this specific talk, I'm going to focus on one case example that we recently worked on. It's for mesalamine delayed release tablet. So in this case example, we use PBPK modeling to support the B evaluation of this GI locally acting product. The question that we tried to answer here was whether the slower release that we saw from the test product would significantly impact the drug exposure compared to the reference listed product. A brief background on the RLD, it's indicated for mild to moderately active ulcerative colitis. The mechanism of action, it appears to have a topical anti-inflammatory effect on the colonic epithelial cells. Based on the label, the bioavailability of this product is around 21 to 22%. The only major metabolite for mesalamine is N-acetyl 5-aminosalicylic acid and is formed by the N-acetyl transferase activity in the liver and the intestine. And this is principally done by NAT1 enzymes. So the PSG recommendation for mesalamine delayed release tablet specifies 3 types of studies. First is a fasting in vivo PK study using a single dose replicated crossover design. Second is a fed in vivo PKB study. Again, same in single dose replicated crossover design. The third study that it recommends is an in vitro comparative dissolution study. So this is a 3-stage dissolution study, which is stage 3 at multiple pH values ranging from 6.5 to 7.5. So for this specific case example that we have, the 2 products were found to be bioequivalent for systemic PK under both the fed and fasting studies. That is, in this case, the drug passed fed PK study and the fasting PK study. However, we found some differences in the dissolution data that was submitted. To look further into the dissolution data, so the dissolution specification is it's a 3-stage dissolution method. Stage 1 is for 2 hours in 0.1 normal HCl. Stage 2 is in 1 hour in phosphate buffer at pH 6.4. And stage 3 is in for 8 hours phosphate buffer, but at multiple pH values ranging from 6.5, 6.8, 7.2, and 7.5. So if you look at the table to the right in this specific case example, what we saw that among the series of dissolution data that was submitted by the sponsor, at and above 7.0, the dissolution profiles for both the products were comparable. However, at lower pH from 6.9 and below, there were some differences between the two products. So the different pH values, usually that the dissolution testing is done over, acts as a surrogate for the in vivo drug release in the GI tract. So in this case, the test product specifically showed decreased release compared to the RLD at pH less than 7. So for this case example, just to summarize, what do we know so far? So we know that the drug product passed the fasting and the fed PKBE study. However, so that means they're bioequivalent for systemic concentrations. However, knowing that we saw some differences in the dissolution at lower pK, the question we were trying to address is that if there are some dissolution differences, will that impact the local pK or the local bioavailability of the drug products? So for that, we took a PPK modeling approach. So here we have listed the general steps for the PPK model. So we were able to obtain the IVPK data, which was used to estimate the disposition parameters. Once we had the disposition parameters, we progressed to the PPK model for the delayed release tablet. To inform that model, we used the 3-stage dissolution data at multiple pH values and used it as a direct input. And we also optimized the intestinal first pass effect to account for the metabolism. And once we have developed the delayed release model, we further validated the model using additional dissolution and PK data that was available to us from additional clinical VE studies. So once we have sufficient confidence in our model, we apply the model to do a couple of things. First, we want to evaluate whether all the series of dissolution data we had, which one is more biopredictive. And once we know or identify the biopredictive dissolution data, we then use it to predict the local amount in the colon and also compare the percentage of drug absorbed in the colon between the test and the reference product, and thereby support the B assessment. So just to go through the model development steps, so the IV bolus data was obtained and we fit it to a 2-compartment model to obtain the disposition parameters. Proceeding to the delayed release tablet, the 3-stage dissolution data that I described before, we used the data at pH 7.2 as a direct input into the oral model to inform the absorption, and the intestinal first pass effect was also optimized to account for the metabolism. And as you can see here, the model is able to reasonably fit the RLD data with reasonable prediction error. So again, we are able to obtain multiple PK data sets, and we use that to validate our model. So we obtained a data set from the literature for the delayed release tablet, again, which was able to be predicted pretty well. And we also had internally available dissolution data sets, additional, as well as PK data sets. And we used it all for the model validation. As you can see, the prediction error was pretty reasonable for all the data sets that were available. So once we have the confidence in our model, the next step we wanted to do is to apply our model. So as I mentioned before, we have a series of dissolution data at multiple pH at stage 3. So what we did is we incorporated all the series of dissolution data into our model and then look at our evaluation metrics in this case, which is the percent prediction error. And we saw that as we decrease the pH or the dissolution data at the lower pH of 6.8, the prediction error was getting higher. So based on that, we concluded that the dissolution profile at both pH 7.0 and 7.2 at stage 3 is more biopredictive to the PK profiles with the prediction error less than 22%. So once we identify the biopredictive dissolution data, then we use that to predict the local amount in the colon and the percentage of drug absorbed in the colon. So for the colon amount, you can obtain a colon amount versus time profile, and then you can calculate the typical or the traditional PK parameter Cmax and AUC for the colon amount versus time profile. And if you look at the simulation results using the biopredictive dissolution data, we can see that the local amount is similar between the RLD and the test product. In addition, we did some population simulation and we showed that the percentage of drug absorbed in the colon is also similar between the RLD and the test product, with the 90% confidence interval for the T by R ratio lying between 80 to 125%. So basically it showed that within the biopredictive dissolution data, the local amounts are similar for the test and the reference. To summarize this case study, the PPK model developed by the reviewers was able to predict the local amount in the colon by incorporating the dissolution data at different pH conditions. We supported that the 3-stage dissolution profiles at both pH 7 and 7.2 at stage 3 may be biopredictive to the local and systemic exposure. The percentage of drug absorbed in the colon is similar between the RLD and the test product. And the predicted amount of mesalamine in the colon is similar between the RLD and the test product. So when we do these reviews, in addition to the PPK modeling, which is the main factor, we also consider multiple factors, such as the dissolution data, the formulation differences, compositional differences between the two formulations, the gut physiology, etc. And combined with all the information, we used to conclude that the test product was low risk for bioequivalence at the site of action for this specific product. So that was a conclusion of my case example. So in our group, we also have multiple GDUFA-funded research focused on GI locally acting product. So we are working on the in vitro BE methods and developing in silico method models for these products. So the goal is to develop in vitro biopharmaceutic data, including solubility and dissolution data for the GI locally acting products, and then use it to conduct model-based virtual BE evaluation in both healthy as well as patient population. Currently, 2 of the grants that are inactive is 2 of the grants that are active is with University of Bath and University of Florida, and Dr. Nikoletta is going to talk about some of the related work in the next talk. Just to conclude, I would like to acknowledge my division members and the leadership for their support and this opportunity. Thank you. This concludes my talk. And
good morning, everyone. It's my pleasure being here again, and thanks to the organizers for the invitation. I'll go on a bit on the GI locally acting products. As Sherin talked before about that case, and I'll show you some work that we have done a couple of years ago. Just trying to, will it be that one? No? The other one? Okay, thanks. So we've done some review of the literature back in 2019. And it's obvious, of course, that the GI diseases, would affect the oral drug absorption. So we tried a bit of identifying different factors that would contribute to these effects. I'm not going to go into too much detail today due to the time, but the point that I want to show you here is that based on these differences that we have in the physiology, we tried a bit of develop some methodology in the in vitro to understand how all these differences would affect solubilization and dissolution of drugs. So based on the differences between patients with GI disease and healthy subjects, We did some identification of the factors that would change really in the composition of the gastrointestinal fluids for patients. Here I'm showing you the case for Crohn's disease. We have done this for other couple of diseases. So here we know that we have an increased prevalence of bile acid malabsorption, in decreased gastric acid secretion, and increased fecal osmolality. So we know that these factors would affect the absorption of the drugs. And for this reason, we took the approach of trying to modify and develop media simulating really these altered conditions in the GI tract of patients with Crohn's disease. And we based our approach really on the current, let's say, media that we use in terms of biorelevant media for fasted or fed state. So just as a schematic, you can see that we took different parameters for the different compartments of the GI tract, and we changed the levels of the bile salts or the pH or the osmolality. And we've used a bit of a design of experiment approach. Of course, you understand that, because of this variability, and we just wanted to understand and to take into account the inter-individual variability in patients with Crohn's disease. We had quite big range of media, but again, we could see a bit of like how different compounds would be affected. It's part of the first part of the study that I'm not going to discuss this today, but we really had a big range of compounds with different properties in terms of lipophilicity and ionization, and we try to identify what would be the risk of altered solubilization in patients with Crohn's disease. So you can see that, in different compartments based on the differences of the fluids in these patients, we could have altered solubilization of ionizable compounds or lipophilic compounds. Now, just to talk a bit of like how I could integrate all this biorelevant in vitro data in modeling. So we use these aspects of solubility in this type of media that we developed. And then we went to do some really dissolution studies under healthy states and other Crohn's disease conditions, and then developing a model. So we worked with budesonides, that is a class 2 compound, and is a locally acting corticosteroid. That is mainly metabolized by CYP3A4 and has quite high protein binding. The formulation used was Entocort. This is a control release formulation commonly used in Crohn's disease. It has a trigger pH, releasing pH higher than 5.5. And these are multi-particulate control release formulations. So we have the gelatin capsule, then we have the enteric coated pellets inside the gelatin capsule. Now, as I said already, based on the aspects of what is happening in the Crohn's disease, so we have changes in the gastrointestinal transit time, changes in the composition of the GI fluids, enzyme abundance, permeation and transporter abundance, and the microbiota. So all of these factors were taken into account in the in vitro and in the in silico models. So for the in vitro, we use this kind of media in the healthy conditions, the classical, media of SGF, FASIF, and FASCOF, simulated the conditions in the gastric, intestinal, and colonic fluids, respectively. And then we have done, the testing for media simulating the Crohn's disease conditions. And as you see here, I have aspects of low and high level. As you remember, I told you that we developed media with different levels of the three main factors. So in this case, we couldn't do really dissolution in all this media that we developed. So we used media that had the low levels of the three factors and media that had the high levels of these three factors. And again, you can see that we did studies with the USP4 apparatus, the flow-through cell apparatus, sequential media changes, and you can see that we have a gastric residence time reduced, taken into account in the in vitro setup. So from 1 hour that you have a gastric residence in the healthy state, we went to half an hour for the Crohn's disease conditions, and we have an increased intestinal phase, small intestinal phase. By the in vitro study, of course, it is a control release formulation, and you can see clearly, with the different healthy conditions or Crohn's disease conditions, we can see from the we can see a bit of like the differences in the profile, and mainly, they would relate to the release of the, the time of the release after the gastric emptying. So then, thinking about the PBPK work, modeling work, we developed first the model for healthy subjects. So we have a clear workflow, and I just as a point of like how you should integrate the dissolution data. We use different approaches, and in this case, or we use the option of using discrete profiles or the triggering pH with Viable. And we selected these ones, and especially for the triggering pH with Viable, the advantage is that includes variability for time that subjects reach the triggering pH in their GI tract. So, and we use this data and trying to understand exactly what is the impact of the input of the dissolution data in the model. And in this case, as you can see, I'm just showing this in the way of our model development, so we could get quite good prediction in both cases, both with the triggering pH and with the discrete dissolution inputs. And I want to mention also here, based on the studies that where we had from the in vivo data published in the literature, we could clearly see that the model captured also the interpatient variability. Now, what is the next step? So we have a healthy subject PBPK model, then we developed the model for the Crohn's disease patients, and we started off doing the parameter sensitivity analysis. And we investigated the parameters of the CYP3A4 activity based on hepatic and intestinal extraction ratio of midazolam, and also the plasma proteins. And I have to mention here that we took also into account the sex difference in this one, as we had decreased plasma protein in female by 44% and 37% in male, and also investigating the gastrointestinal transit time and gastric pH. So with the parameter sensitivity analysis, as you can see, these factors selected, you can clearly see that we have an impact on the Cmax and the AUC by varying the CYP3A4, the hepatic or the intestinal one, also the albumin levels. And in this case, with the gastric pH, of course, the impact is clearly shown when we reach the triggering pH of 5.5. Remember, this is the pH that we have release of budesonide from the formulation. And now we go and we develop the model, the PBPK model for the Crohn's disease patient. We have the low levels and the high levels of the main factors identifying affecting the absorption. So would be the enzymes and the albumin. And we input now the dissolution data that we generated from the biorelevant in vitro approaches. So here we can see the performance of the Entocort formulation in patients with Crohn's disease, based on the in vivo data available, and you can see that with the green line we simulate the healthy simulation. So if it was healthy subjects, whereas with the blue and red lines are the two different levels of simulating the Crohn's disease patients, and you can clearly see that we get quite a good prediction. So this is a bit of like, yes, the work that we had started with this one, and now we continue working with the grant that we got from the FDA, or for these parameters, these formulations or other formulations. And to us, it's important to understand that how you can use these alterations in solubilization and dissolution in the appropriate in vitro setups, and then the integration in the modeling. And of course, I would like to thank people who worked with me on this one, that it was at that stage funded from the European Union, the Pearl projects, and also, people from Sertara and SimSip for working in this case. Thank you.
So very good morning to everyone. Good afternoon and good evening to the folks overseas. First of all, I would like to thank CRCG and FDA Organizing Committee for inviting me to speak here today. So I'm going to talk about the PBPK modeling for evaluating GI locally acting product from industrial perspective. So this is my standard disclaimer that this presentation reflects my own view and does not reflect COSET's views or policy and does not reflect any official policies of regulatory agencies. Here is the agenda for my today's presentation. We'll begin with the brief introduction of PBPK modeling, followed by its application in drug development. After that, we will talk about the PBPK modeling on GI locally acting product. Challenges involved, key consideration for the PBPK model development, method development and validation criteria for the PBPK model, regulatory perspective, and conclusion. So let's start with the brief introduction on PBPK modeling. So as we all know, PBPK has a long history and this has evolved slowly over time. In past few decades, there have been a lot of research happening in this area. Regulatory bodies are also encouraging the approach. This is very important tool for drug discovery and new drug development. So what is PBPK modeling? It is nothing but a mathematical modeling approach which contains multiple compartments to demonstrate the ADME, absorption, distribution, metabolism, elimination of the drug products, and its metabolite. This also used to evaluate the exposure of the drug in the target organ after the administration of the product by considering the rate of absorption and disposition in the organ. This diagram here is a conceptual PBPK model which represents physiological and biochemical parameter and its extrapolation for human health risk assessment. Let's go over the application of PBPK modeling. Basically, this model can be useful for new to generic drug development through the product life cycle. There are some application of PBPK modeling for new drug product development as well as generic drug product development. For new drug product development, PBPK is mainly used to select the initial dose for pediatric population, dose optimization for the clinical study, also to evaluate drug-drug interaction. Whereas for generic product development, modeling is mainly used for bioweaver purposes like bioweaver in lower strength, bioweaver for comparison between test and reference, to establish the dissolution BE space, to evaluate the clinical impact of product critical process parameter and critical material attributes. Recently, regulatory agencies have published guidelines emphasizing the importance of PBPK modeling in generic drug development. Let's talk about the PBPK modeling for GI locally acting product. Here, active drug undergoes dissolution in GI tracts. So bioequivalence for GI locally acting product is very challenging, as drug reaches to the site of action faster than it reaches to the systemic circulation. B study with clinical endpoint is insensitive to detect formulation differences, and it also requires greater number of subjects. For GI locally acting product, the rate and extent of drug absorption at site of action is mainly governed by dissolution and the transit of the drug along the GI tract. In vitro dissolution is very important to identify the potential in formulation differences between RLD and generic product. Also, PBPK model integrates existing knowledge about the physiological parameter and drug properties. Pharmacokinetic differences between test and reference can be predicted by the use of verified PBPK model. As we all know, there are several challenges involved in developing GI locally acting product. Let's discuss few of them. Local drug delivery. Precise drug delivery to target the site in GI tract is very challenging. Measurement of the drug concentration at the target site is difficult. So systemic drug concentration may not reflect the local concentration. Variability involved in GI physiology among individuals and even with the same person over time makes the development very challenging. The inherent complexity involved with GI tract also makes the development difficult for the GI locally acting product. Food interaction can significantly alter the absorption and release of the drug in GI tract, and formulating the product regardless of the food effect can be difficult. Optimizing appropriate dosage from GI locally acting product are technically challenging as well. Here, in vivo study can be challenging due to the practical consideration. Patient variability can impact the performance of the product. And finally, developing GI locally acting product is cost and resource intensive. As we discussed, the challenge is how PBPK model can address these challenges. PBPK model can incorporate anatomical and physiological variability to represent inter-individual and disease-related variability. By adjusting this parameter, the model can simulate the GI tract variability across different patient population. PBPK model can also allow the division of GI tract into various compartments, which evaluates the product behavior at the target site within the GI tract. PBPK model can also account for the factors such as formulation characteristic, dosage forms, and drug release kinetic to predict the local concentration profile at target site. This model also can help assess the impact of food on product absorption by considering parameters such as meal composition and timings. This model can also integrate in vivo, in vitro data to enhance its accuracy. And PBPK models are based on the mechanistic understanding of drug behavior. The more information we can put, it improves the predictive capability of the model. Let's go over a few key consideration of PBPK model of GI locally acting product. Target GI site. We have to specify the target GI site where we want our product to work. This can influence the model structure. We must use all the literature and background data available to optimize the model accuracy. We have to gather all the data available related to GI tract, incorporate the data from healthy individuals as well as patients with GI disease. Characterize the drug property and evaluate how this property can vary in different parts of GI tract. Perform the sensitivity analysis for model's robustness. Model should be able to demonstrate in vitro, in vivo correlation of the product. We have to consider the variation in GI physiology in different patient population. And there should be a model validation and verification process to assess the model performance and its accuracy. Let's briefly discuss about the PBPK model development criteria. Model development requires multiple input. It requires detailed understanding about input parameters and the model structure and its intended purpose of the use. Depending on the use, input parameter can be used. For models for drug-drug interaction, the parameter for metabolic rates are critical, whereas models to study absorption, the formulation and drug release parameters become important. As discussed, it is always recommended to use literature data to predict the behavior of the model. During the PBPK model development, in vitro dissolution method development should be initiated. And as soon as the dissolution method is developed, it should be validated against the available information. This is an example of flow diagram of model input parameter required to develop PBPK model. Here, this is a typical software example of simulation plus for modeling, which shows the different feed to incorporate the available information. Let's talk about the validation. After the development, it is very important to validate the PBPK model. PBPK model validation is a crucial step. Relevant in vivo data will be used to validate the model accurately. Model parameters like drug-specific parameter and physiological parameter needs to be validated. Perform the sensitivity analysis to evaluate the impact of variation in model parameter. We have to consider both internal and external validation to evaluate the model robustness. We have to evaluate the prediction error. Prediction error requirement for IVIVC is 10 to 20% based on the internal and external validation. However, the prediction error for PBPK modeling is challenging due to the physiological as well as formulation variability. Use of systemic PK data to verify GI lab PBPK model. here we have to consider to disqualify the GI locally acting product PBPK model that fails to predict the general characteristic of systemic PK profile. PBPK model can help evaluate whether the systemic exposure reflects the local drug delivery, especially through the shape of PK curve. For example, PBPK model predicted a correlation between systemic mesalamine plasma PK and its gastrointestinal distribution. These findings are implemented in the B assessment in product-specific guidances. Let's discuss a regulatory perspective on the PBPK modeling. The regulatory authorities are showing interest in the application of PBPK modeling to support the drug development and regulatory decision-making. They have also identified the potential of PBPK model to enhance the understanding of drug behavior in GI tract. These are the published guidance reference from US FDA and EMA. These guidances include the information for developing models, development strategy, data requirement, validation, and verification approaches for PBPK modeling. These guidances are also providing a highlight for the application of PBPK model development for several different purposes like clinically relevant dissolution specification setting, quality risk assessment, supporting post-approval changes, reducing number of clinical trials, and etc. So to conclude the presentation today, model verification for GI locally acting products serves as a key step in using model to inform regulatory and drug development decision. Verifying such models can be challenging, mainly attributable to difficulty in obtaining drug concentration at site of action. And advancing technologies to generate relevant in vitro and in vivo data that directly and indirectly reflects local drug delivery, leveraging systemic PK or using additional data from relevant drug product can collectively serve as weight of evidence approach for model verification. At lastly, I would like to acknowledge my team member who has helped me to make this presentation. And finally, I also would like to thank CRCG and FDA for giving me this opportunity. Thank you.
Hi, I'm Chung Tsang. I'm the other moderator of this morning session. Thank you for all the great presentations. So we'll start the panel discussion. Would the speakers come to the podium? And we also have invited a few more people to join our panel discussion. They are Dr. Xiaojian, the Deputy Director, DB2 from FDA, Dr. Rebecca Moody, Pharmaceutics Reviewer, DB, Dr. Amin Rostami, Professor of System Pharmacology at the University of Manchester, Dr. Romi Singh, the Senior VP at Sun Pharmaceuticals. We have Mr. Harshil Shah and Dr. Romi Singh joining us virtually. We have prepared some questions for this panel discussion. Let's go through each prepared question, and we'll ask some panel members to give their comment. Now, because of the limited time, I will ask each panel members to perhaps make sure that your comments will be brief, so that we'll have enough time for the attendees to ask questions at the end. Okay. The first question is, based on your experience, what are the latest advances and utilities of using oral PBPK absorption modeling to support generic or new drug regulatory submissions? For this question, I'll ask Dr. Wu, Dr. Heimbach, and Dr. Jiang to give the comments. First, Dr. Wu.
Yeah, thank you for the question. Based on our experiences and our review in the regulatory submissions, we saw some actually case examples which successfully support a tentative approval of the drug product. This is for using PBPK to set clinical relevant particle size specifications. And the other examples actually I mentioned in my presentation include using PBPK to assess the impact of non-comparable alcohol dose dumping in vitro testing and the non-comparable dissolution across different strands for supporting the biowaiver. And some other applications include using PBPK to justify the BE study design, like using single-sex BE subjects, study subjects, and also, as Dr. Sherin Thomas mentioned, using PBPK to evaluate the BE for GI locally acting products. That's based on my experiences.
Okay, thank you. Dr. Heimbach?
Yes, I think the latest advancements include the use of physiologically based biopharmaceutics modeling to support drug product quality. We are seeing more and more applications where we're justifying the dissolution specification or product particle size distribution specifications. And I think in terms of the model tools themselves, we see that there is inter-subject variability that's always included, but also intra-subject variability can now readily be parameterized for virtual bioequivalence trials. So it's basically parameters such as gastrointestinal pH or transit time or bile acid concentration that can be included in the latest PBPK absorption models.
Thank you. Dr. Jiang?
Yeah, so from the FDA review discipline perspective, we have been seeing that using a modeling approach to address internally and externally some of the B-related question, for example, Tmax issue, TLAG, and there are some cases we require dissolution to be comparable, where we see different dissolution compared test and reference. There's also approach using modeling and simulation to address the difference. And also, we also seen that there are some situation for the lower strengths, which is not proportional, and then the applicant that using modeling approach to address the to request a waiver. So I think we see the common issue is to validate the model. For the B perspective, we are not technical for PKPD modeling, so we ask our expert in DQMM to help the evaluation. But I think in general, we see there's a concern for to get a validated model. And I think also it's very challenging.
Okay, thank you for all your comments. Any panel who want to give a brief comment before I go to the next question? Okay, the second question is, what are the challenges and possible solutions for developing patient-centric dissolution quality standards using PBPK modeling? With that, I'll ask Dr. Ashraf to perhaps give a first comment, if you will.
Yeah, thank you. Yeah, the first, I would say is the first challenge is mimicking the in vivo dissolution profile using in vitro dissolution methods. As you know, no apparatus is ideal. This is a work in progress. Many companies are trying to modify, etc. These kind of apparatuses. Some commercially available apparatuses are also now available. On the other side, the effect of food and disease conditions, etc., are the conditions which are not easy to replicate while developing dissolution methods and doing in vitro dissolution testing. Then developing the models, the lack of the incomplete information from the clinical studies under disease condition and food conditions, etc., is the second difficulty and challenge which has to be overcome in order to develop successful models to predict patient-centric dissolution specifications.
Well spoken. Next, I'll ask Dr. Rebecca Moody to perhaps give a comment.
Yeah, thank you. Yeah, I echo everything that was just said. I think the main challenge with, using PBBM to develop patient-centric dissolution specifications or clinically relevant is establishing that IV-IV link, the in vitro in vivo link. And that could be due to, not enough clinical studies with PK variant or variant formulations. There's also the question about the dissolution method for input is QC or using a physiological relevant media. And I think, part of the other issue is just that there's lack of global acceptance and harmonization and, minimal transparency between industry and FDA, with regards to the data and what is expected of them. So I think solution is, more workshops like this, more communication, and early communication between industry and sponsors to provide so they can get feedback on their model and modeling approach. And a big one, again, for building the IV-IV link is, to start that modeling approach early in the product development and not use it later on as a Band-Aid for when it fails QC specification.
Thank you. Any comment from other panel members? Yes?
Yes, I think, our biggest challenge is to actually change our philosophy of the way that we are seeing in vitro data. Everything that we heard today with regard to dissolution, for instance, they were binary. People were doing, in acid 1.2 and in, 7.2, and then even they know the GI tract is a continuous change of the pH. We are trying to actually to use these two dissolution to fit what we are seeing until we come back and get comfortable with actually different philosophy of breaking this dissolution on the basis of what we are seeing 1.2, 7.2, but also filling the gap between those two, knowing that in GI tract there are different shear stresses, different pHs, different bile salts. We won't be able to actually address patient-centric element.
Good. Very interesting comments. Any other comments before I go to the next question? Okay. The third question.
I have a comment and I have a question here.
Could you identify yourself? I have trouble hearing you. So
can you hear me? Yeah. So I'm Ahmed Zidan. I'm from Office of Pharmaceutical Quality. I'm working in FD Labs. So I have a comment and a question here. For patient-centric quality standards, I think the ideal situation is just to bring the gastrointestinal tract to our lab, right? So this is the ideal scenario, but do you think we have all the facility and all, I would say, the instruments that can capture all the physiologic conditions that's happening in the GI tract, regarding transit time, regarding emptying rate, regarding pH gradient, and others? So I believe there is no single dissolution method that can capture all the physiologic condition. So this sensitivity analysis and screening studies to understand what is the critical parameters from the physiological part that can affect really the bioperformance is the key here in choosing which dissolution method that may be biopredictive, right? So this is my comment, and it will take me to the question. And for the locally acting drugs, we have seen that USP4, the flow-through cell, is used as one of the biopredictive dissolutions. So what is the rationale of using USP4 or the flow-through cell for locally acting drugs when you don't have any systemic exposure? USB4 has its own limitation, right? Regarding smaller volume, for example, in the cell, grid, like maybe irrelevant flow rate, but it can mimic sometimes the systemic circulation. However, for locally acting drug, it may be like more elaboration is needed for this.
Okay, any response to this? Go ahead.
Probably I can give you my opinion on this. I would agree with you that, there is no way definitely to replicate everything from an in vivo situation to an in vitro setup. In general, my approach is that sometimes, with in vitro setups, you can identify a bit of like risks. So for me, I really like a bit of like using a way of like screening. I will never tell you that this is the best method, that is the only method, but several cases, when we study, an approach and then we use different types of setups, again, you can understand and better also what could affect more your formulation. You can get a bit of like better way of characterizing the critical attributes. You can get a bit of a way which factors would really affect and be a risk really for this formulation. And then I think that nowadays compared to 20 years ago, that all these were not at this stage is now. By linking this with PBBM, for example, I think that there, you can use and you can get a better even understanding. So I don't think that anyone here is absolute that this is the best and all this, but I really believe that with a range of some in vitro tools and then using your modeling to understand and get this even parameter sensitivity analysis and seeing where you will have an issue, then you can go back and you can have and refine your methods. And just to say a bit of like, because you mentioned about the USB4, I mean, again, for any approach, for any formulation, in any in vitro setup, there are also these more complicated systems like TEAM or, apparatus and all this. But I don't think, sometimes I think, you can just start from a simple approach and then you can go on based on the complexity that you have to deal with. USB4, in my opinion, has some advantages in a way of like the sequential media changes and so on. But I will not say that is the only approach that will be used. So sometimes, just a single approach, again, because we have now the chance to combine more with modeling, you can use a single profile even from a USB2. Even though I have to say that several times, USB4 to me, it gives some aspects when you talk about precipitation and all this, it's much easier really to define some of this. Thank you.
Just one more question, before we go on to the next question. Please, Walt.
So Rob Leinberger from the Office of Generic Drugs. So I have a question solely for the industry members of the panel, okay? And I want to understand how far we are from, as part of every oral product development, saying that your internal expectation is, I'm using PBPK modeling in my routine product development because there are so many different places that it can be used in your product lifecycle for different specification settings, biowaivers of different strains, different dissolutions. How far are we from the point where you as the industry members, this is irrespective of what you submit to FDA, I don't want that to be the issue. I want to say internally, how far are you from the point where you are sort of routinely expecting that your development programs all use this? And certainly I'm interested in Tycho's perspective from the brand development, but also the generic industry perspective on that question.
So if I understand, the question was how confident are we in industry to use PBPK across the drug development process? And I can tell you that probably only a very small amount of PBPK or PBBM applications reach regulatory agencies, because these days the tools are there. And the data is also readily available. So for example, if a company makes a decision to make a modified release formulation as part of early drug development, you would want to make sure that you have looked at the models to make sure that you have an ideal candidate for modified release formulation. The same thing goes if you are in a competitive field, and typically we are for polywater soluble drugs. For example, in oncology, we would like to have the knowledge whether or not an enabled formulation is needed. Enabled formulations are more costly to develop, so we use PBB and PBPK to inform these types of decisions. So I'm confident to say that for all projects, we use PBPK models in all cases. There's very few projects I can think of where we're not using PBB and PBPK as part of either a formulation decision or in other applications such as prospective TDI assessments.
Thank you. Maybe I'll ask Dr. Singh to perhaps give his comment from a generic perspective. Dr. Singh, are you online? Yes.
Romi, you're muted.
Maybe I forget industry too.
Yep. We can see you.
Hello, am I audible? Here you go. Yeah, okay. So, from a generic perspective, we are usually using IV-IVC correlation, PK-Sim, for routinely all our developments, and also selection of dissolution media. And most of them are internal, and in some cases we do submit it to the regulator. But in terms of application of PBPK modeling, we attempted it for submission to the regulator in one of the cases, and that was to establish the dissolution safe space. And incidentally, it was for one of the locally acting drug products. But we were not successful. So ultimately, I think we went ahead as per the PSG, and we defined our dissolution safe space as per the expectation of plus minus 10% of the BE batch. So we are doing it, but in terms of meeting the regulators' expectation of validating as well as still meeting the PSG requirements, it becomes a lot of challenging in terms of I think the current concept wherein we can have the conversation with the regulator before we even attempt doing it would perhaps enable us to be much more online with the regulator's expectation than like doing the full scale, submitting it, and then getting into to and fro questions with the regulator. So currently, in terms of submission to the regulator, our case, we have submitted only one case for establishing dissolution safe space, but we were not successful. Instead, we agreed to the plus minus 10% of the BE batch.
Okay, thank you for...
So maybe I can make a small comment from the industry side, because I worked 15 years in a major pharma. In 15 years, we tried 3 times. And only one success we had, which was approved level A IV-IVC using PBPK. And those were early stages actually of even developing PBPK. So that was only one success in a major pharma in 15 years.
Okay, very good discussion. Dr. Polly, can I ask you to hold on your question? We want to go to more questions, as many as we can, because of time is running out. The third question I want to ask the panel is, when do you think the community should consider harmonization opportunities for all drug products, including the modified release product, using PBPK modeling and simulation approach? What are these opportunities? For that, I ask Dr. Almeida to give a comment.
Thank you. So I think when we talk about harmonization, we tend to think about the process at ICH, which is a very formal process. But actually, I would dare say that harmonization starts before that. And what we're doing here is laying the basis and the foundations for future harmonization by approximating the way that we think about these topics. So you mentioned modified release. As I mentioned in my presentation, this is going to be a new topic adopted by ICH. I guess in the same way that we did for M13 and immediate release, the group of experts who will work on the modified release topic will have the opportunity to consider where the knowledge is at the time that they start to work on this. And if they see fit, they could consider it. But I will not speak on behalf of that future committee that is still not set up.
Any other comments? Yes, Dr. Rostami?
Yeah, the difficulty of not having harmonization, when you look at from the industrial perspective, which I am not from, but I'm in touch with most of them, is that if there is no harmonization, the effort that goes into producing a PBPK model for absorption or whatever that you are doing and our experience in DDI and other areas becomes to complete absolute waste, because that another, let's say, regulatory group is not going to accept it. So everybody defaults to the sort of the worst case scenario, saying why I am doing this when the X and Y and Z are not going to accept it. Let's just go and do whatever we have traditionally do. So to move away from this tradition, I think it is essential, because every business in industry these days is global. It's not just, to FDA or just to EMA or wherever, because it is global. If there isn't, that harmonization, I'm afraid, sort of this is not going to pick up.
Fully agree. Okay, well, let's go to the next question. What are the key considerations for adequately developing, validating PBPK modeling for local drug exposure in the GI tract of GI locally acting products in healthy versus patient population? It is a very important question. So I have a few members that perhaps want to chime in for this discussion. First, I'll ask Dr. Thomas to give her comment.
Thank you. So there's a couple of things we need to consider. First of all, like the dissolution input that you put into the model itself. So if you have like a series of dissolution data, just to put some thought into what's the most biopredictive among them, and which will reflect your in vivo behavior as closely as possible and potentially predict your in vivo outcome as accurately as possible. Second factor to consider is the local bioavailability. So systemic concentrations are important. So for the model development and validation, you do use a systemic concentration, but at the same time, in these kind of products, as we discussed before, the local site of action concentration, whether it be the early on in the GIT or the colon, whichever it is, it's also equally important, and it should be considered in the model development and validation as well, and to support the BE. And the third part was, if you're doing the modeling in patient population, then there's more additional factors to be considered. As Dr. Fotaki's presentation, she covered several of the physiological and biological changes that happen in disease states, such as ulcerative colitis and Crohn's disease. For example, the pH changes in the GIT, the microbial changes, as well as the fluid compositional changes. All of these factors would have to be considered and potentially incorporated in your model to have a more effective prediction in a disease state.
Okay, Dr. Fotaki?
I think that I don't have really too many to add what Sherin said, but exactly, I think that the difficulty that we have mainly is for the locally acting ones, and as you see, I carefully selected the compound that has some systemic absorption, so I can, systemic concentration, so I can at least validate my model. I think that sometimes the difficult parameters is when we have local concentrations, and I think there, we try a bit of work even with animal models sometimes and try to extrapolate this to humans. But I see that more on the aspect of the validation of the model. As I say, for the ones that have no systemic absorption, it's going to be challenging, but the perspective there really is if we can have, from the work that we are currently doing with Sherin and Fang, is mainly, for some combines that we have at least some information, so we can develop these models. But I'm thinking if we want to see this like generally, for all the GI locally acting products, it will be challenging. And then also just to say in terms of the microbiome and all this, there are several cases, again, that we take into account in the modeling, and we have developed this platform with Simulations Plus that we work in this model. Yeah.
Okay.
Thank you.
Dr. Rostami? Want to give a comment?
I would say one of the actually opportunities that we have got when we go to the patient, side of the thing is the differences that we are getting in the variability. And this was discussed, in the workshop that we had obviously 1 month ago here. I think we have not used the recovery of the variability as a way of validation of the models. We are so much just focusing on recovery of the central tendency that we forget recovery of the variability also can help with the verification of the models. I think this is definitely an opportunity, because we know, as Dr. Fotaki was talking about, sort of the variability within patient community that they are receiving is going to be very different than healthy.
Okay. Mr. Shah online, any comments?
Yeah. So as other panelists are explaining that, first of all, it is very, very challenging. And, there are few main considerations which I already discussed in my presentation while developing the model, where we should look at the as much as data available to optimize the model accuracy. Also, when we are developing and validating these models, we must perform the sensitivity analysis. And model what we develop at last should at least demonstrate the in vitro in vivo correlation for the locally acting products. So these are the few key considerations we must look at it while we are developing this GI locally acting products.
Can I add a caution with regard to, the last comment on the sensitivity, again, for particularly benefit of those people who did not attend the 3-day workshop. Sensitivity analysis, we have to be very careful, because if we are looking at parameter scanning, which is one parameter changed when all the other things are fixed, this is very misleading. Majority of the parameters that we have got in GI tract, they are connected to each other. If you are changing stomach emptying, it is impossible to actually assume the pH of the top part of the GI tract would be as it was in the previous set of the model parameters. We have to make sure that the correlation between parameters when we are doing sensitivity analysis are considered. So a word of caution there, but there are enough publications now that people can actually look into.
Last, we'll ask Dr. Singh to give your comment online. Dr. Singh? Yes.
Yeah. So two things I would want to speak here. So there is a recent yet unpublished article from Center of Applied PK Research in UK with SimZip, and the model drug is budesonide. And they have compared the healthy state versus disease state, as well as the in vitro and the in vivo bioequivalence. And there has been significant discordance found between the in vitro and the in vivo bioequivalence at the upper GIT, which is more sensitive to formulation differences, and as compared to the lower GIT. But when it comes to the modeling done in disease state, the results are different from what we have in the healthy volunteers. So obviously there is a lot of complexity involved. Now, coming from a generic industry perspective, and if we go through the PBPK modeling approach for future submissions, so establishing it both in healthy volunteers as well as in a disease state may have its additional challenges. And for example, rifaximin, another locally acting drug, 2 strengths, different indications, different disease conditions. So it would add to the challenges that we have in terms of adopting PBPK modeling and proving the validation, the internal and the external validation. So obviously it would be challenging for the generic industry. But yes, in case we are partnering with the regulator as part of the complex ANDA, I think we would want to tread that path and understand more.
Okay, thank you for all your comments. Because of time, I'm going to stop asking questions. I'll open up to the floor, and maybe we have time for a couple of questions. Yes, please identify yourself.
Yeah, hi, Sunit Shukla, Boehringer Ingelheim. So I have a more pragmatic question about this approach, and it relates to the locally acting products. So, for example, what Sherin presented in terms of the model, you have an indication where, let's say, for mesalamine, if you have to go to pediatric population, then it's a weight-based dosing then, in that case. And you have to modify based on the age group here. And there the physiology comes into picture. And from PBPK perspective, even if we have a model in place, how confident we are in terms of going to pediatrics based on that model, especially for the dose consideration.
Any response?
So, yeah, I mean, I haven't really thought about that before, but I think the confidence depends on the purpose of the model. But if you are talking about applying it to pediatric, I think then the validation and the model development, there are certain factors that are more important as to, like in my specific case example, the local concentrations were predicted. We did not have any observed concentrations that were available to validate the local concentration. The model was validated based on the systemic PK. So if your purpose is to apply it into a different population, then I think additional, as much as observed data that we can get to validate the model, to increase the confidence in the model. At the same time, the physiology. I mean, PBPK, you can develop the model for pediatric as well, right? So you will build a model with the purpose of applying it to a different model, population, but at the same time, I think the validation will be definitely more important in that case. But in this case, the application, that example that I provided was to just support the BE. We already had PK data. We knew it was systemically bioequivalent. And then we also knew that the dissolution was very similar at the higher pH, and we know the formulation design itself. So we had a good understanding of the risk involved. So it was more used as a risk assessment tool. So in this case, I think we just wanted to predict the local to provide confidence that the risk was lower. So, but in your case, I think we would require more observed data. More confident.
So it's more applicable for the generic formulation, where
exactly,
you know the formulation, and then you can probably do some extrapolation. But if you go on the new drug side, that may not be applicable. You need more clinical data.
Exactly.
And I think at the end of the day, the question is, can you use this model to do pediatric dose justification, which seems still not that easy, using, even if you have a very good model in place.
Can I just add, to the discussion on this one, on the back of the question? I think actually we have to think of the question in this way. Are all different conditions for which we want to use the model, we will be in a position ever to actually to come up with what is so-called validation? Because this is not going to be possible. In many of these cases, we know that the model cannot be basically actually validated in any form apart from indirectly later on. Going back to what actually Dr. Romi Singh actually referred to, that unpublished data. In fact, that's published. Chun Yan Han, it was in the list that actually was shown earlier on by Dr. Almeida. But these are the kind of things that we are not going to be able to ever to actually study. And the equivalent case would be, I invite everybody to look at what our colleague from OCP, actually Dr. Joe Grillo recently and the colleagues published with regard to having the full circle. This is the story of rivaroxaban, which J&J was submitting with regard to renal impairment and the drug-drug interaction being different in renal impairment patients. They did not have any clinical data, and yet they did the PBPK, they put some warnings in the label without any clinical study. And after 10 years, using real world data analysis that I am sure OGD people also have been using to look at, generics versus, non-generic, etc. They showed what the warning that they had given already on the basis of model was correct. Those people who had renal impairment and they were using inhibitors of 3A4 and PGP, they were at higher risk of bleeding. They did not do any PK study to show what the actually PBPK has shown is true or not, but indirectly they could show that the side effects were higher. And had they not have that in the label, imagine how many more of the physicians and pharmacists that in the comfort of blank that you have got in the label would have done, sort of that combination, and we ended up with more patients being actually experiencing that side effect. Therefore, the question should be framed in a way that, in many of these cases, we know we cannot do the studies. Shall we leave it blank? Shall we make the assumption, for instance, adult to pediatric is always the good thing? Or shall we actually fill in the gap using PBPK modeling with some real world data evidence and so on later on backing up our decision?
Okay, I'll give last comment to Rob.
Yeah, so again, another comment on the GI acting products. So first I want to, one of the things is echo Amin's comment about the fundamental things in that recent paper that he published, right? Looking at what's the local concentration. That's the key variable for our bioequivalence, but it's never going to be measurable, right? And so if you think, well, and I think in FDA our challenge is sometimes people get the idea of validation. Well, I can't use a model until you show me that it predicts the actual data. And I think this is a really good example. It's never validatable at that level because that's just an inaccessible measurement, but that's the key quantity that you're interested in. And I think that's a sort of fundamental challenge. And, for us internally in FDA and how we internalize, either use the models and our internal stakeholders to convince people of the validity of the models, right? There's a lot of the mentality of, to show me that it predicts the data, and then I'll believe that it predicts the data, and then you can use it instead of collecting that data. And that doesn't move things forward. And this is a good example of that. But again, another comment on the locally acting GI products. I think this is a really good place where the PBK models are going to be very valuable for the industry. And as Xiaojian and others on the panel can indicate, for these mesalamine products specifically, there's a lot of stuff that happens in the studies. If you follow our product-specific guidance, sometimes you end up with ambiguous results. Oh, one parameter failed. Does that mean it's not bioequivalent? Or do we have to use some kind of judgment? Or there's a little bit of how do you put together the dissolution data with the PK data to come to a decision? And the model, and these things get consulted a lot. In my office, spend a lot of time analyzing these. PBK models can be extremely helpful in thinking about, is this observation that we had in this sort of ambiguous situation, what does it mean? And there you can use things like I mentioned in that paper, like you can say, well, what effect does this have on the local concentrations that I'm really trying to ensure when I'm trying to put together the weight of evidence. So that's, again, for the generic industry, for these more complicated situations, the value of developing that model ahead of time can really help you interpret that ambiguous data. Certainly if you do every study that's in our product-specific guidance and they all perfectly exactly pass, right, probably your product will just be approved on that. But in more complicated situations, that just doesn't always happen. There's cases where there's ambiguous results. That's a challenge for our reviewers, but we also, it's an opportunity to use a more scientific and quantitative analysis as part of that evaluation. So just really, that's one of the values for the generic industry of having the PBK models built up. So you understand, like, oh, how much risk is this? And then you can begin and be involved in that discussion. And I think that's something that can lead to the faster approval. So thank you all for the additional time for that comment.
Thank you, Rob. With that, I'll close off the panel discussion this morning. Just to remind you that there will be a roundtable discussion this afternoon for further discussion on similar topics. Okay, with that, I want to thank all the panel members for your participation this morning and all the great comments you gave. Thank you.